Perfect. Thank you, Kitai, and thank you everybody for joining today. I'm going to share my screen. Let me know if you see over here. Okay, can you see my screen okay? Excellent. Perfect. So, as I said, thank you again for joining this week. If you remember last week, Tom started with a brief introduction into really what stem cells are, how we use them in the field, and maybe a brief introduction in what we're going to be covering throughout the next several lectures. But today we're going to be focusing a little bit more on the what we'd like to consider as the upfront or the upstream part of our workflow when it comes to reprogramming or somatic cell reprogramming. So we're going to talk a little bit about, what reprogramming is. We're going to give you a little bit of a background and discuss the different Yamanaka factors. And then we're going to go into the different reprogramming technologies. And we're going to focus a little bit on the Sendai reprogramming, as it seems to be the most prevalent method that most folks use nowadays. And then finally, again, I'll introduce the concept of characterization PSCs that we're going to see throughout the rest of our lecture series as well. And same as as last week, please, if you have any questions, if something is unclear, feel free to ask questions in the chat. Stop me and ask any questions live as well. We're happy to take any and all questions throughout too. Okay, so you've seen the slide before from last week, right? Where Tom introduced you to what pluripotent stem cells are and kind of where they come from, introducing the concept of or the true definition of what a stem cell is, right? So if you remember, drop it in the chat, right? Tom mentioned there's two properties that we typically consider for stem cells to be truly stem cells. And the first is that they must self-renew to a certain degree. And the second is that they must differentiate into other cell types, again, to a certain degree. And this certain degreeness, if you will, if I might use that term, really dictates where they are in terms of their pluripotency status, going from totipotency all the way to unipotent or multipotent cells that can eventually differentiate into terminally differentiated tissues. Right. And Tom again introduced this concept of how we use these cells, right? We typically start with a somatic cell culture. We figured out a way to reprogram them, which is what we're going to be talking about today. And then eventually we're going to get to how we culture these cells, characterize them, engineer them, and finally differentiate them. But before we begin, right? Again, I'm referencing a lot of what Tom said last week, just as a reminder. But we did mention that there's different types of stem cells based on their source, right? And at a very early days in stem cell research, most of us were employing embryonic stem cells that were harvested from the inner cell mass, as you see over here, plated down into culture and used to study these cells in vitro. What we're going to be talking about is induced pluripotent stem cells today, which is slightly different, but they're quite similar to embryonic stem cells as well. Now, before I start, and again, Tom did throw this slide in here. Tom did mention that one of the breakthroughs throughout stem cell history is the cloning of Dolly, the sheep. Now, I want you in the chat to kind of venture a guess as to why that is. Why is cloning part of stem cell research? Drop what you think is in the chat. Let's see if somebody gets it in there. Maybe I'll give it a 30 seconds to a minute, see what folks are thinking. Nothing so far. Maybe nobody knows, but that's still okay, right? The reason why I'm mentioning it to cure disease, okay, that's a good guess. So the reason why we consider this to be truly a hallmark of stem cell research specifically is when doing the cloning, right? The idea was to take the nucleus from one cell and put it into another, and then take that zygote or cell that was made and transplant it into a surrogate sheep of a different coat, of a different color, right? And when Dolly was born, it resembled the features or the hair coat of the original donor of the nucleus and not necessarily of the surrogate, right? So what this told researchers back in 1996 is that the cells are not as static as we thought they are, right? That the nucleus is almost like a center of command, right? Where the cell can dictate to the what it's supposed to be doing by instructions that can potentially be given from the nucleus itself, right? And this sparked the idea of how we can potentially change the cells based on the information that's coming out of the nucleus, right? Which sparked a little bit of the reprogramming kind of conversation. And reprogramming, if I were to define it, right, it's the process of reverting somatic or adult cells back into their pluripotent stem cell-like state, where they behave like embryonic stem cells, right? So think about that for a second, right? With Dolly, we were able to understand that cells could potentially change their fate based on the genetic content that's within their nucleus. So, several researchers, most notably Shinya Yamanaka and Jamie Tomlinson in Japan and here in the US, respectively, were thinking, well, what if we applied that logic to cells and we force them to express certain factors that are known to be expressed only in pluripotent stem cells or in embryonic stem cells, right? So they went on this screen basically where they were trying to figure out which factors that are expressed in the embryonic stem cells that if induced in a somatic cell like fibroblasts like you see here would potentially start this cascade of events that can lead into a shift in state, right? And this is exactly what happened. So what Shinya ended up publishing, and Jamie as well, is they found that the expression of four factors, right? OCT4, SOX2, CMYC, and KLF4 is enough to induce a terminally differentiated fibroblast cells. And this work originally was done in mice, but it was then later replicated in human. So these factors that are now termed the Yamanaka factors are able to induce this cascade inside of these cells that would start a transcription cascade that would change the state of the cell. And if you think about it in a very simplified way, it's almost like reversing time, right? Where a terminally differentiated fibroblast can reverse time and start to act more like those very early embryo-like cells, right, that are in the inner cell mass, and potentially give rise to everything in your body, right? Which allows us to use these cells like somebody mentioned in the chat for to treat diseases, for drug screening, to even model human disease in vitro, right? So this is how kind of the concept of reprogramming started, right? So the expression of these four factors we knew was enough to induce in some cells, at least at the time, this signaling cascade that can reverse back time. Again, I'm saying that in a very simplified manner. But if I were to ask you, well, how do you do that, right? How do you get these cells to express these four different factors to reprogram and turn into induced pluripotent stem cells, as we call them, right? And this is where you have to think about the different reprogramming methods. And before we get into the technologies themselves, I know that this is an eye chart of what you're looking at, but it's almost intended to be, right? These are the different delivery methods in which we can force cell types to express certain transgenes or proteins, right? And at the very top, you'll see the very first distinction in the delivery method is whether it's viral or not, right? So we can use viral particles to deliver a cargo of a particular transgene, or there's also non-viral delivery methods like transfection, electroporation. There's even newer methods like LMPs and things like that. But if we were to go down to both of these delivery methods, whether you're talking about viral or non-viral, The main way we like to think about our different reprogramming methods is whether they're integrative or not. So again, I'm going to ask folks to participate in the chat a little bit and kind of tell me what you think that means. What is integrative versus non-integrative reprogramming methods? I'll give it a little bit in the chat. There you go. Alexis came in with the win. There you go. Nice. Um, awesome. Actual insertion. So you guys are absolutely right in the chat, right? The when we refer to integrative methods, we're referring to methods where that transgene that you're trying to deliver into the cells ends up incorporating into the own cell's genomes. Whereas with non-integrative methods, it's more of a transient expression that's later lost, right? So that your transgene itself is not affecting the genome of the host cell that you're introducing it into. And this is an important concept because, I'm sure a lot of folks are very familiar with various different ways in which you can deliver cargo or transgenes into a cell. But we've used, things like lentiviruses and retroviruses quite a bit. But those can be integrative methods where whatever you're trying to deliver ends up integrating into the host cell's genomes, right? Which, if you think about in this context, can be a little problematic, right? Because if I'm going to be inserting, in this case, four different genes into the genome of my cell, I'm going to have to worry about where these genes land, right? Are they going into a spot where they can create a mutation? Are they causing a problem in an enhancer element or a promoter sequence, right? And for this reason, we tend to go with non-integrative methods in the field a little bit more often than not, right? Where the most two commonly used methods at the time is deliver using the Sendai virus, where those four reprogramming factors are used and delivered via the Sendai virus, and we'll talk about that in a second. Or the more traditional methods where those transgenes are delivered as plasmids, right? That end up being expressed but not necessarily integrated into the genome. Although there has been some reports that there could potentially be a small risk of integration with episomal methods that I thought I'd mention. And we won't go into it too much today, but on the up and rise is instead of using viral methods, using our mRNA as a modality to deliver those transgenes, where instead of delivering them in plasmid format that could potentially last and integrate into the genome, as I mentioned, the mRNA reprogramming would actually just provide the mRNA template of those genes that you're trying to express, and the cell can do most of the work there, but then these mRNA transcripts will usually get lost because mRNA is not very stable, right? Its half-life is much shorter than DNA is. We'll talk a little bit about mRNA reprogramming, but not as much as the other two methods. Now, I'm sure now you're pretty much all confused, right? And you're like, okay, there's way too many ways to do this. How do I figure out what reprogramming method works best for me, right? And when trying to decide this, I usually like to think about four different things in order to set the stage of, well, what do I need to consider, right? And when I'm doing my reprogramming. The first and pretty much the first thing I go to at least is the safety of the method. And safety here can mean two different things, right? It can mean safety for the researcher, the operator, right? Whether you're using virus or you're using non-viral formats, there could potentially be different hazards associated with it. As an example, I'll give usually the Sendai virus is considered a BCL2 containment level contaminant. At least that's the case in our facilities. But that's not always the case in every single lab, right? It's going to depend on your institution and your regulations as well, right? In certain parts, in certain labs in Europe, I know that Sendai is not considered a BCL2. It's considered differently. So again, the safety of the method itself is something to consider for the operator themselves, but also for the cells, right? different delivery methods can potentially cause different rates of aneuploidy in the resulting cell, right? So you'll see, for example, with retroviruses, because they can integrate potentially, there's a higher risk of aneuploidy in these reprogrammed cells compared to sendai, mRNA. And again, I did mention with the episomal factors, there's a slight risk of integration as well. So that's the first thing I would consider, right? The safety of the method. The second thing is the consistency, right? How consistent am I, or am I able to generate clones every time I use this method? Especially if you're considering picking multiple clones, or if you're working with really precious samples, right? Where you can have one go at this, and you only have enough for one go, right? You'll see that the reprogramming methods have gotten decently consistent, I will say, across time. But one method that's still being optimized right now is mRNA reprogramming. This is relatively new, and I still believe that the success rates and the consistency will continue to rise as we, further optimize these mRNA reprogramming workflows. But at the moment, they're a little bit less consistent than the traditional methods because we have a little bit more experience with them. Now, the third thing I would also consider is workload, right? How much work are you incurring by picking one method over the other? And this is specifically important too if you're planning to generate multiple cell lines or multiple clones, right? Where traditional methods, have been standardized, but again, newer methodologies like mRNA reprogramming tend to be a little bit more time consuming because they require multiple different transfections across multiple days in that very initial stage of the protocol. And don't worry, we're going to talk a little bit more about this, but I just wanted to introduce the concept of workload and time as another factor you should consider. And then finally, another really important consideration is the somatic cell type that you're trying to reprogram. Are you trying to reprogram, fibroblasts, blood cells, specifically any type of other cell type that you're looking for? And now I worked with folks at UCSD that used to reprogram placental cells, right, or mesenchymal placental cells from the cord. So the cell type that you're reprogramming is important. And the reason why I say this is most reprogramming methods right now will do a decent job of reprogramming proliferative cells, but whether they're adherent or not can make a potential difference. Right? And as an example here, I just wrote it out at the bottom. You see that blood-derived cells with mRNA reprogramming, at least the first several iterations of those mRNA reprogramming kits could only be differentiated after you've established an endothelial progenitor cell line, which takes around 2 to 3 weeks, which could increase your workload, potentially also shift the cells as they become more clonal during your selection. So these are just a bunch of things to consider, right? And if I were to give you some advice around, how to pick your method, typically what we look at are the two most important things that I like to frame at least is around safety and efficiency of your method, right? So if you're looking at all these different ways in which you can deliver these reprogramming Yamanaka factors into the cells, what you want is basically a method that's highly safe, but also highly efficient, that sits in this top quadrant over here. And I just listed, this is just an old slide, but you see that the three methods highlighted in red here are three methods that we offer, for example, at Thermo Fisher specifically. Now, other considerations that I think should also be mentioned in when you're reprogramming your cells is a little bit around where you obtain your sample from, right? The comfort level or invasiveness of the procedure that of obtaining your sample. Right now, skin fibroblasts are quite easy. They're quick punches, but blood draws could potentially even be easier, right? And this is specifically true. Again, I've dealt with this with multiple different researchers, but with infants, for example, a skin biopsy might be a little bit too invasive, but if they're already sampling blood, they could potentially use that as a source for their cells in order to start the reprogramming as well. And when thinking about that and thinking where you get your cells or your original sample that you're trying to reprogram, also think about the sample size, right? How many cells are you able to get? Because we have to work with a certain number of cells in order to initiate that reprogramming protocol. So if your sample does not give you enough cells, then maybe you have to think about a different source or a different cell type that you're looking to reprogram. The another big thing that you see pointed out here is the ability to reprogram that specific cell type, right? I briefly alluded to this, but um, for most reprogramming methods, reprogramming non-proliferative cells is extremely difficult, right? You want the cell to be a little bit more proliferative, which allows it to change its DNA conformation that allows for these a little bit more quickly so that you're able to start this reprogramming cascade by introducing those four transcription factors or four Yamanaka factors. Right, so these are just a couple of other considerations to keep in mind. Now, um, before I start into the different reprogramming methods, any questions so far in the chat or anything anybody would like me to address or elaborate on a little bit further? Tom, anything in the chat worth mentioning or calling out as well that you've answered? Please let me know too. We can kind of cover that
Yeah,
with each other.
the one question that came, there are a couple of interesting questions that came through. We talked a little bit about in the chats susceptibility for different cell
Hmm.
types and things that can impact that. And the response I gave especially was cultural longevity and cell type behavior,
Hmm.
right? But if you have anything else to add there, certainly feel free. And if not, no worries.
No, it's a great call out, and it actually leads into what I said. Like the susceptibility is for me as well as related to how how well these cells are equipped to reprogram, right? So their previous culture methods, even cells that we've reprogrammed before, right? Like PBMCs or blood cells, there are factors we'll talk about in a second that you have to consider before you start the reprogramming to kind of gear those cells up and get them adapted enough, if you will, get them ready for this process because it's an intense process, right? You're asking the cell almost to reverse back time, right? Again, I'm going to use this simplified, metaphor, but it's not an easy thing to do, right? It's almost uprooting everything, erasing entire epigenetic signatures and starting from scratch, right? So you need to prep those up and get those cells ready. We'll talk a little bit more about that, but good point though. Sweet. Anything else? Why actively divided cells are preferred? Is anything to do with plasmids entering inside the nucleus during actively dividing cell types? So it's a good question. I think I don't believe we know exactly why, or we haven't determined the exact mechanism. But during the cell cycle, right, there's these epigenetic exchanges and DNA conformations that allow for a little bit more accessibility to the DNA. So I think a part of that is making sure that you're able to access the DNA itself, like you've mentioned too. Would you comment a little bit more about reprogramming of placental-derived MSCs? So, yeah, with the MSCs, what we were doing at the time was taking cells from the cord and plating those down to derive mesenchymal stem cells. And with those cells, then after a while and after we've characterized them, then we would reprogram. And the idea there, one of my colleagues at the time, who's a brilliant scientist, but what she was doing was she was collecting MSCs from different patients with a known outcome, right? Specifically, she was looking specifically at hypertension during pregnancy or preeclampsia. And knowing the end state, right? She would take those MSCs and reprogram them into PSCs to figure out if we could figure out what would be the cause during the initial placental development that might be linked to those preeclampsia cases as well down the line. I hope that answers some of that. Are there ways of increasing consistency of reprogramming using mRNA? There are, Jordan. So there's definitely ways, and folks have been working on them a little bit. Again, we'll briefly touch on this. So no worries there. And I see Tom and Keita already also chimed in. So let's keep it moving just for the sake of time. But from this point on, I hope that you appreciate that the process, although technically simple, right? If, hey, we want to deliver and get these cells to express four transcription factors. And by doing so, most proliferative cells will revert back from that somatic state into a pluripotent state where they are able to give rise to more, most cell types at least. That's the logic there. And there's different ways to do it. One of the ways to do it, as I mentioned, is to deliver these factors using plasmids, right? And at Thermo, we have a way to do this with our episomal kit, where we deliver those reprogramming factors that you see, OCT4, KLF4, and SOX2 on this plasmid, and CMYC as well here, right? But you also see a bunch of other things, right? Like Lin28. This is a dominant negative form of P53 and EBNA transgene as well. And the reason for this is even though those four factors are sufficient to reprogram the cells, right? The reprogramming efficiencies when they were first described were very, very low, right? I'm talking about 0.001%, like on that scale, right? And since then, we have figured out different ways, and I think Jordan, you asked this question to optimize the delivery and sorry, not delivery, to the optimize the reprogramming efficiency by adding a little bit more factors in. So Lin28 and the dominant negative 50 P53 form increase reprogramming efficiencies with these episomal factors. And the idea is with these episomes, right, we can deliver these plasmids either via electroporation by basically zapping the cells, where the electricity will create holes in the cell wall by which these plasmids can enter, and then the cell will heal the cell wall afterwards. Or, classically, we can also use lipid transfection, with reagents like lipofectamine to deliver these plasmids, right? And the idea, again, here is that this is a non-integrative a reprogramming method, right? So these episomal vectors should remain in the cytoplasm, and they will replicate every time these cells divide. But then, due to DNA replication or partitioning, these episomal vectors will be lost at around 5% per cell generation or per cell cycle. So typically, after reprogramming, you'll see that within 5 to 10 passages, these transgenes or these plasmids are no longer detectable in your cell lines that you've reprogrammed, right? I will say that there is a caveat here that there has been some work to show, or some work in the literature to suggest that in very rare instances, there has been integration based on sequence homology of some plasmids into the DNA. So just a quick thought to consider. The other method I mentioned is it's very commonly used is by employing the Sendai virus, right? So instead of delivering the reprogramming factors as plasmids, right, or genes within a plasmid, in this case we're using the Sendai virus, which is a rodent parainfluenza type 1 virus. And I know some of you are thinking, are we talking about this again? Like viruses? Maybe it's just me and I'm scarred from COVID, but one thing to point out here is that this particular virus is non-pathogenic to humans and it's an mRNA virus that replicates in the cytoplasm, right? So For for these purposes, it is considered to be a safe way in which we can deliver cargo into cells without harming us as researchers as well. But with that kept in mind, and you obviously all know that viruses replicate within the cell and they actually use a cell machinery to get, or they hijack the cell machinery in order to replicate their own genomes and eventually end up killing the cell because they create more viral particles inside, right? In this case here, right, part of that viral genome has been altered so that you're hijacking the cell's machinery not to express viral gene, the virus itself, but also your transcription factors. And in this case, we've made sure to delete or most folks have made sure to delete the F gene that's part of this viral genome backbone. And the F gene specifically is responsible for assembling viral particles. So by doing so, even though we're using a relatively safe virus, we're also trying to ensure that we're not recreating infectious particles that can keep reinfecting cells after that initial transduction or after you initially started, right? So the Sendai method, like I mentioned, is really widely used. It's the kit that's currently available in the market is the second iteration of that kit that was launched in back in 2023. And it has three different Sendai vectors, right? It comes in these three tubes. The first Sendai vector will carry what we call the KOS vector. These are the KLF4, OCT4, and SOX2 genes, right? And then there's one other Sendai vector that will carry the c-Myc transgene. Then you'll notice that there's a third one with an additional KLF4 vector. And the reason for that is because this is a second iteration of this kit, we figured out that the relative expression of those four different genes is also important. So not just their presence there is important, or the fact that these proteins need to be there to start the reprogramming cascade. It seems like their relative expression or the relative abundance of these proteins can also impact reprogramming efficiencies. And so you'll see from the first iteration of the kit to the second iteration, the reprogramming efficiencies of either fibroblasts or blood cells has increased dramatically, right? And again, we'll talk about this in just a second. In so far with both methods that I've described, and pretty much with almost all methods at this point, you can reprogram your cells to set up PHC cultures in two different formats. And we'll talk about this actually in the next lecture. But you can do these reprogramming protocols so that you can set up feeder-dependent cultures where your pluripotent stem cells that you see, colonies that you see over here, are basically sitting on a bed of mouse embryonic fibroblasts as supportive cells that we call feeders. Or you can set it up as in order to establish feeder-free cultures, where this layer of mouse embryonic fibroblasts that you see here is actually replaced with extracellular matrix proteins or coatings directly onto the plastic as well. And don't worry, we'll talk about both different systems in the upcoming lectures as well. So as one of the more commonly used methods, I'll focus, like I said, a little bit more on the cytotune or the Sendai reprogramming specifically today, and I'll talk a little bit about reprogramming fibroblasts as well as reprogramming blood cells as well, or PBMCs. But this is the general protocol, as you see here, the schematic for reprogramming protocol with the cytotune, the Sendai virus, right? It takes around 3 to 4 weeks, where you would start off by culturing your fibroblasts that you're intending to reprogram for about 2 to 3 days up until they reach a certain confluency. After which this is where on day zero, this is where you add your Sendai reprogramming vectors, right? Those three vectors that I mentioned that contain the Yamanaka reprogramming factors. And you'll maintain these cells in that fibroblast media for about 7 days. And during those 7 days, you'll actually it's really cool. You'll start to see the cells themselves shift in morphology and go through this mesenchymal to epithelial transition where they become a little bit more cuboidal. And at around day 7, the cultures can get quite dense. And so what we do at that point is we lift the cells off of the plastic, these cells that are, undergoing this cascade or this reprogramming cascade, and we replate them onto either MEFs or these or this or a matrix, like I mentioned, in order to start initiating or start supporting the pluripotent stem cells that are starting to emerge, right? And once these cells attach, you'll see that by day 9 to day 10, these colonies will start to form and they'll start to divide, and you'll see them. And I'll show you some pictures in just a little bit. And these colonies that start to form at day 10 or day 9 or day 10 right here will continue to grow over the next, 2 weeks, to by the end of, 21 days, we'll start to see these really hopefully healthy looking colonies that we can pick, transfer over into a subsequent passage, and then continue to propagate as our pluripotent stem cell population that we've reprogrammed, right? But before I get into all of that, one thing that I really wanted to touch upon is sort of MOI, right? Or multiplicity of infection, as we like to call it. And the reason why I'm referring to this, anybody who's worked with viruses, you'll know that we use this term quite a bit, MOI, which indicates basically how much of the virus am I adding, right, to my cells, right? What's the multiplicity of infection? How many viral particles do I want, or viral vectors do I want per cell, in order to achieve a good delivery outcome, right? And in this case, we at Thermo, with the Cytotune kit, have done a lot of work to provide some recommendations on the amount of virus for each of those three vectors that you need to add, right? So we have an MOI of 5 for the KOS vector, an MOI of 5 for CMYC, and an MOI of 3 for KLF4 vector, right? And what this basically means is when you're doing your reprogramming, right, you'll plug in the recommended MOI, or if you're planning to use more or less of the virus, you can decide that on your own. But you'll multiply it with the number of cells that you're transfecting, and then you'll divide that by the titer of the virus, which is provided to you usually by the manufacturer, in order to get a volume in microliters that you were to add to your cells in order to initiate your transduction, right? Or for your cells to get transduced and get these genes delivered into your host cells. Now,
Omar, can I add one thing here?
please.
We
Yep.
do sometimes have to modify or optimize the MOA a tiny bit. We often do give call outs in the protocols that we have, etc. But these numbers are by no means absolute. I believe, and I don't have it in front of me, Omar, so correct me if I'm wrong, but isn't it KOS and CMYC has to be 1-to-1, and the others we can sort of tweak a little bit as needed in order to optimize for a specific cell line or cell type?
Cor- Correct. Yeah, correct. For the most part, we try to keep at least like the ratio the same, particularly for the KOS and the CMYC. Sometimes the KLF additional KLF4 vector can be kind of manipulated or optimized a little bit further as well. But I will say, Tom, you brought up a really good point of why do we sometimes change or deviate from the recommended MOI, right? There are certain scenarios where, with what we like to refer to as hard to transduce cell types, or potentially samples coming from patients that are at risk of not reprogramming. And I'll elaborate a little bit further. So I'll give you an example. Typically, samples obtained from older patients are not always, but on average, a little bit harder to reprogram compared to younger patients, right? So sometimes you'll see, especially with folks doing disease modeling, if they're getting their sample from an 80-year-old male, for example, right? That they might have a harder time reprogramming, even though if it's a cell type that is known to be, reprogrammable, right? So they might want to increase the amount of virus they add in order to ensure that the reprogramming factors are indeed truly expressed at a high level in that cell in order to drive the reprogramming cascade, right? Sometimes too, with precious samples or very difficult to try and do, or cells that are not very proliferative, again, you might want to increase that MOI in order to achieve a decent delivery and reprogramming results. Good call out, Tom. And actually, a good segue into, we've done a lot of legwork, at least at Thermo here, and there's a lot of literature about it too. So not just work from Thermo internally, but of how much virus you should be adding for different cell types that you may be working with, right? But if you're working with a particular cell type that you don't necessarily know, where to start, right? There is no solid literature on how much Sendai virus to add or something, similar to that. There are tools that are at your disposal that can help you solve some of these questions where this is a good example where in this case the Sendai virus is carrying a GFP target as opposed to the reprogramming factors. Now, this Sendai vector will not do any reprogramming, right? But what it will allow you to do is figure out at what MOI your cell type is best transduced at, right? So you can use tools like this in order to also figure out and streamline your reprogramming before starting, and because it's a 3 week protocol, right? So you don't want to start and then have to wait 3 weeks before you can figure out an answer, especially if you're optimizing and you're not really sure how much of the virus to add in the beginning. Right? But this is what it looks like, right? So if I'm showing you, these are pictures or representative images of fibroblasts before the reprogramming or on day zero, right? So you'll see in this case around 80 to 90% confluent. Some folks recommend as a range of confluency ranging from 30% to 90%. Again, depending on the kinetics of your particular cell line, that might need to be optimized, but it's a good starting point. If your cells look like this, they should be good to go, and you should be able to start initiating your reprogramming, right? When you do, at around day two, you'll start to see that those cells are transitioning, right? They're losing that mesenchymal morphology and starting to become a little bit more cuboidal, as you can see over here, right? And this will continue as these cells grow at around day seven, right? And if you remember day seven for the fibroblast protocol, this is when we would lift the cells off and replate them. And by day nine, you start to see these clusters of cells that look slightly different, right? Can anybody, or can you guys see that? And how these look very different to these surrounding cells, right? It's a little bit more prominent here, but as these small clusters start to grow, right, you'll notice that they make these colonies with these defined edges and particular morphology that we'll talk about in a second that is referencing the morphology of pluripotent stem cells specifically. Now I know images are good in a Tom knows this really well about me. I tend to make up expressions, and English is not my first language. I'll preface it with that. But I tend to make expressions and make up expressions in my head. And I was going to say that I think there's an expression that says a picture is worth 1,000 words, but I don't know how much a video is worth because I have a video for you of this entire process, right? So this is before I start the video, this is a time lapse video of the reprogramming process from day zero all the way to day 20 to 21. And you'll see, individual cells and how they continue to behave in culture, especially after that replating on day seven. I want you to pay attention around day nine, day 10, when you start to see these colonies pop up. I'll press play here. I hope this works. Let me know if it doesn't. But here you'll see we start off on day zero with our fibroblast culture, and we added in our day zero again our Sendai virus. These cells will continue to grow, and you'll start to see them change morphology by day two, day three, right? As you can see over here. And at some point, they'll become very confluent, right? Don't be scared. They're supposed to. We don't recommend actually dissociation before day seven. But once you do, as you see that cut in the video, that's when we dissociated. But when we plated those cells down, right, these colonies start to emerge, right? They look very, very different than the surrounding mesenchymal cells. And those actually are your pluripotent stem cell populations or your pluripotent stem cell colonies. Now, I want you to notice here that the not all the colonies are the same size, not all of them are the same shape, and not all of them have the exact same boundary, right? This is a in the center here, this is a really nice reprogrammed colony. But if I were to go to the side down here, right, you'll see that there was a colony emerging, but maybe its edges aren't as defined as this right here, right? And I want you to pay attention to that because we'll come back to it in just a second. But uh, each individual colony that you see is, um, in theory, right, a single cell that was able to reprogram, right? It's a cell that received all of the reprogramming factors and was able to drive that signaling cascade in order to initiate reprogramming, but more importantly, also not just initiate it, but continue and finalize that reprogramming process. This is a concept that's maybe a little bit weird, but we do see some colonies that initiate reprogramming in the beginning, but then they start to differentiate pretty quickly afterwards. And sometimes you even have colonies that halt, right? They start to grow, but then for some reason they just stop growing completely. And the reason why I'm mentioning all of this is after the reprogramming is finished, right? When you're looking at taking those cells, specifically the pluripotent stem cell ones, and carrying them over into the second passage or continuing to propagate them in the next culture vessel, right? The idea is that you want to try to pick the healthiest cells that you can, right? Because these cells just underwent a big change, right? They again, I'm going to use this again. They reversed time, right? And went back to their pluripotent stem cell like state. So you're going to try to pick the colonies that look the healthiest, and we'll give you some parameters on how to choose and how to distinguish between what is a potentially a good colony versus a not so good colony. And before I do that, just a couple of things I wanted to also share. There is, as I mentioned, the Sendai virus or the Sendai method is widely used. It's actually one of the more efficient reprogramming methods currently, and for a whole bunch of different cell types and a whole bunch of different species. This is really, really interesting work, but there are several companies, and also there's some work that was going on in the San Diego Zoo, so not too far from a lot of you, where they were using the cytotune kits in order to reprogram cells from different species to try to save particular species of animals from extinction, right? Some of you may have heard this, but there was efforts to help or derive pluripotent stem cells from the white rhino. There was only two white rhinos in the entire world that I think were both in the San Diego Zoo. There's also it was in the news not too long ago, but there's a company that's uh, that did some reprogramming in order to in efforts to try to bring back the woolly mammoth or the dire wolf, right? Some of you may have seen some of that on the news as well. So there are already a lot of use cases here, and part of the reason why that's true is because of the versatility of the Sendai virus, right? The Sendai virus specifically needs the sialic acid receptor in order to properly integrate or transduce a cell, but those receptors are abundant on most cell types. So they're easy to transfect for this particular virus, making it a decent or a good approach to take. Now we talked, and I showed you this video about fibroblast reprogramming. we can do similar things with from other sources, right? I mentioned another very commonly used source for reprogramming cells is blood. And so typically what we do is we take whole blood and we separate it either by positive selection or by density gradients. You see here, this is a histoplaque, so it's at a particular density, and you add anticoagulated blood and you spin it down so that the different layers go into the different densities where you can have the plasma on the top, the buffy coat here, which will contain your PBMCs or your peripheral blood mononuclear cells. These are cells in your blood that contain things like your CD34 positive hematopoietic stem cells, for example. And you'll see down here, these are your erythrocytes and your granulocytes. Typically, we don't reprogram those cell types. Well, one, erythrocytes don't really have a nucleus, right? But they're also non-proliferative, so they don't make for very good reprogramming targets. But from these PBMCs, right, you can even, you can reprogram whole PBMCs there, or you can further isolate cells from this layer, right? Because T cells, B cells also can be derived from PBMC or can be isolated from PBMC. So depending on what cell type you were working with, there are protocols that already exist. So if you need any help, please reach out. We can point you in the right direction. And this is just an example of one of those blood-derived reprogramming protocols with the Sendai kits, right? Where you'll see that it's very similar to the fibroblast reprogramming in a sense, maybe towards the later end of the protocol. But in the beginning, for PBMCs, we typically have a culture period. You see these negatively noted days as sort of days we like to keep these cells in culture as they acclimate to conditions, kind of related to the susceptibility question that was asked earlier as well, right? And once these cells are comfortable enough, then we think that they're ready to go in terms of number, they're looking healthy. We can start the transduction by adding our Sendai viral vectors on day 0. And in this protocol, you'll see that blood cells, because they're not really attached, they are actually in suspension culture, grown normally, that by day 3, they're going to be ready to be plated back down onto the extracellular matrix. And in our experience, we've seen that PBMCs tend to reprogram a little bit faster than fibroblasts. So by day 14 to 16, we see these really nice colonies that are ready to be picked for most clones. Again, don't be surprised if for whatever reason you do not end up getting a clone for every single time you reprogram. The reprogramming efficiencies are still relatively low. We'll discuss that in just a second, but they're consistent. So for the most part, you should at least be getting 1 or 2 clones that you could work with potentially. Now, another thing I wanted to mention is there's a really good Nature Biomed paper that I like to refer folks to that describes different reprogramming factors and methods, whether they're integrating or not, the reprogramming efficiency. Kind of, it does a nice job of relatively comparing the effort and the cost of each method and sort of what factors are used during. I highly recommend for folks that are considering doing reprogramming work themselves, at least take a look at this resource, potentially similar resources that you can find in order to, like I said, just figure out what is the best approach for you and what would work for your needs specifically. And on that note as well, I will mention that because this course is also done in collaboration with CERM, I wanted to specifically mention how we can also take some of this work into the bedside or the clinic, right? So what we described today is the Sendai reprogramming kit, the Cytotune kit with the vectors that you see right here, right? We talked a little bit about this. But when we're doing work that's related to the clinic, or where work that is intended for patient use, right? Think about what these cells are intended for, right? Tom mentioned this last week, but a big promise of the field is that we can use pluripotent stem cells as a repair kit, right? Where we have access to this, cells that can renew indefinitely and potentially, if given the right clues, can give rise to every single cell type in your body. Can we use these cells to put back into patients to make them, healthy again, right? To treat disease. But if we're going to do that, we have to have considerations on how we approach this with patients, right? Compared to how we do it in just a lab setting. So for this reason, I wanted to briefly mention that there is versions of kits, and we'll talk a little bit more about this towards the end of our lecture series, and going into the clinic. But there are these reagents that are intended use for clinical applications specifically. At Thermo, we have a brand of products that we call cell therapy systems. So this is just an example of some of those reagents that are almost fit for purpose for these clinical applications. The reason why I'm mentioning any and all of this is in this version of the kit that's geared towards clinical application, instead of the c-Myc vector, right, which is one of the classic reprogramming Yamanaka factors, right, that we talked about, we've replaced it with an L-Myc vector. And that's because c-Myc is a proto-oncogene that can potentially drive proliferation of these cells forward, right? And in a clinical setting, we also have to worry about whether these cells are able to transform, right? Especially with cells like pluripotent stem cells that have the potential to give rise to anything. We don't want to risk the development of a tumor, for example, right? So we try to de-risk some of these kits or some of these reagents, right? Additionally, you'll see that in this clinical version of the kit, there's no BSA in the viral dilution buffers. And BSA is albumin serum, bovine serum albumin, which is derived from bovine, from cows. Right? And we try to, again, in these clinical settings, reduce any animal origin components that goes into patient-related workflows as well. So just a quick call out there. But very similarly to the research use version of the Cytotune kit or the Sendai kit, you can reprogram a whole bunch of different cell types with this cell therapy version of the kit. And we talked a lot about what reprogramming is, the kind of topic behind it, the workflows. Didn't really mention to you the reprogramming efficiencies themselves, right? And some of you may be looking at me right now and saying, you've talked this whole big game about how we've improved stuff and things are moving forward, but I'm looking at numbers that look very low here, right? Who agrees with me or who doesn't agree with me? Let me know in the chat, right? But a common question that we get is, well, you're saying that the Sendai kit is one of the most efficient reprogramming kits, but I'm looking at the slide here, right, and saying the highest you can get is what, 1.6% reprogramming efficiency. But if you think about it, we've made a really leaps and bounds because again, the original reprogramming efficiency described in Yamanaka's original paper was 0.01%, right, or less than 0.01%. And so we've gone orders of magnitude higher than that as a field. That said, also think about the other side of the coin. I mentioned to you that every colony that you see, at the end of your reprogramming protocol is essentially a single cell that reprogrammed. If every single cell did that, you'll have a very hard time figuring out which cells or which clones to pick, right? And because of subsequent work, we know that we want to work with clonal and as opposed to heterogeneous populations of cells. So having a slightly reduced reprogramming efficiency isn't always bad because you don't necessarily want every single cell to reprogram. You want the more competent cells that are the healthier ones that end up making this, and you want to even screen them afterwards too, right? So just something to keep in mind. Another thing that might jump out at you from this slide is the difference between the blue bars and the green bars, right? So the blue bars represent the RUO or the research use only version of the Sendai kit that we've been talking about, while the dark green ones represent that clinical version that I just mentioned. And so you see this dip that is in the reprogramming efficiency is very likely due to that c-Myc to L-Myc vector replacement, right? Again, we've touched on this before, but proliferative cells tend to reprogram better, right? So as these cells are starting that cascade, if you have a gene that's able to promote proliferation a little bit better, you'll probably increase your reprogramming efficiency to this degree, right? So this is part of the reason why you see this dip in reprogramming efficiency for the clinical version of the kit. Now, with the reprogramming protocols, right? You'll notice that there is at the later ends of whether you're reprogramming fibroblasts, like you see in the top schematic, or blood-derived PBMCs in this case, like you see at the bottom schematic. There is media that's specifically used in the later half of the protocols that are very specific to pluripotent stem cells, right? These cells, if you remember from Tom's talk, are a transient population of cells, right? And so what does that mean? It means that in the embryo, these cells do not last, right? So in your body as an adult, even as an infant, you should not have any pluripotent stem cells. You might have some adult stem cells or adult stem cell types, but pluripotent stem cells specifically are a transient population of cells. They don't last. So they're in the embryo and then they differentiate. So when we want to keep them in culture, or as we're trying to get them to establish, right, we're trying to get them to emerge in this case, we have to make sure that we use media and reagents and growth factors that will support them, right, that will maintain them in this state, right? And one media that we use here at Thermo is called Essential 8. We'll talk a little bit more about this media and what it means, but there's different media systems that you can use in the later half of this protocol, right? Essential 8 is what we call a lean media. It's not necessarily, it doesn't contain a lot of different factors compared to other pluripotent stem cell medias that you can find. But it's what we've seen is that it does support the best reprogramming efficiency compared to other media types, right? So this is one thing to keep in mind. Um, in terms of not just the media, but the matrix itself, right? I mentioned that you could either reprogram on a layer of mouse embryonic fibroblasts or on an ECM or matrix supported proteins. So which matrix you actually coat your plastic with also matters in terms of reprogramming efficiency. What we found is coating with a laminin based matrix like laminin 521 tends to give you really good reprogramming efficiency, especially comparing to other basement membrane extracts like Geltrex or Matrigel, or even more defined matrices like the truncated vitronectin. Don't worry too much about this. I know these terms can or these media systems and matrices sound a little confusing right now. This is what we're going to cover next on our next call, so no worries there. Before I move on, I wanted to briefly again just touch upon mRNA reprogramming as another non-viral method that's really up and coming in terms of figuring out how do we deliver those reprogramming factors into our cells. So um, if you remember that chart I showed earlier of like safety and efficiency, mRNA reprogramming was very promising, right? In terms of safety, mRNA transcripts are transient, right? So they don't last very long. They don't tend to integrate into the genome anyway. So it gives us a really nice approach of, hey, if I were able to deliver these mRNA transcripts into the cells, I can get a very quick and easy way for these cells to translate these transcripts and then make the correct proteins that will start the cascade right away, right? That said, the first iterations of the mRNA reprogramming kits, They use synthetic mRNAs encoding the classical reprogramming factors, right? So OCT4, SOX2, KLF4, and c-Myc, along with Lin28, which is shown to increase reprogramming efficiency. Um, the interesting thing is, as I mentioned, the half-life of mRNA is quite short, right? So especially compared to DNA. So in order to maintain the expression of these factors, the protocol is required for daily transfections, right? 12 to 16 daily transfections. Not only that, but we also needed to add things like B18R in order to neutralize interferons because with cells, once they see free-floating mRNA, they tend to think, hey, there could be something wrong, right? This could be a pathogen. So they might activate innate immunity and degrade these transcripts. So we needed to add like stabilizers, if you will, to keep these transcripts from being cleared out. And so what these protocols look like was something like this, right? Where you plate your cells, get them ready, and then you do your transfections for 12 to 16 days, which which worked and was compatible with adherent adherent cells like fibroblasts, but didn't really work very well for suspension-based cells like from blood per se. The second and third generations of these protocols are starting to become a little bit better, right? So there's improvements that are constantly being made. I can't honestly speak to this. I mean, we don't offer it at Thermal, but I will mention to you that they're still taking a similar approach in it. But as you can see now, there's a little bit less of a burden on the cells. So they're adding a little bit more immune evasion factors like E3, K3, as well as B18R. They add, I think, some microRNAs as well to increase reprogramming efficiencies, and the number of daily transfections there is lower. Now, this is one thing to keep in mind, right? Transfections in general are not, they are tolerated by the cells, but they're not ideal. So if you can minimize them as much as you can, that would be the way to go. And we'll talk more about this when we hit the characterization piece. But let's say you've done your reprogramming, right? Now you're all reprogramming experts. Everything should be good to go. Um, how do you tell what's going on, what's happening here? So, in terms of the features or the morphological features that you're looking for, there are very distinct things that I would call out here, right? Notice how the cells in this colony are very compact. This is a hallmark of PSC or pluripotent stem cell morphology, where they like to be in very, very close contact with each other specifically, right? And if you zoom in on this, you can see that very, very clearly. The spaces between the cells is very, very small. Another thing you'll notice is that they'll have these defined, hopefully round, refractive edges. It almost looks like you're shining a light from behind it. That's because the edges are slightly beveled. And that's again a good sign of PSC morphology because these cells like to attach to themselves as opposed to their surroundings. So a rounded colony morphology that has that refractive edge is something that we look for. The third thing that you want to look for as well, especially under higher magnification, is this higher nucleus to cytoplasmic ratio. Pluripotent stem cells tend to have really large nuclei compared to their cell body. And maybe it's a little bit hard to see. Maybe if I can zoom in a little bit here. But you'll see that the nucleus makes up a very good chunk of the cell's body, right? One other thing that you'll notice upon me zooming in here is that there are multiple prominent nucleoli. And again, some people freak out when they see this, but don't worry. This is again another hallmark of PSC morphology that we look for, and it is actually desirable. So once you identify, the colony by morphology, and you say, hey, this looks like a good clone or good colony that I need to pick and propagate so I can move these pluripotent stem cells specifically and start to culture them in the subsequent passage, right? The second thing that I would recommend doing, although not everybody does this, is to use tools in order to identify or label your pluripotent stem cell population specifically compared to your surrounding cells. And so there are these live stains that you can use with cell surface markers that are specific to pluripotent stem cells. In this case, you'll see that there's a live stain, two live stains in this case, actually, one labeling TRA160, which is a cell surface marker commonly used to label pluripotent stem cells, while another live stain here was used to label the fibroblasts specifically with CD44. And the idea is you want to pick the red cells and not the green cells, knowing that the red cells are the ones that reprogrammed, and your green cells are fibroblasts that still maintain that CD44 marker, right, which is a fibroblast marker. Now, before I get into the next step, I will mention that, and I did again allude to this briefly, not all colonies are going to look this beautiful, right? Some colonies might have this one side looking great while another side is looking slightly more differentiated, right? Some colonies, if two cells that have both reprogrammed right next to each other and they are starting to develop, might start to emerge next to each other and eventually merge, right? So not every colony is going to look this good, right? But with morphology as well as with, some staining, some live staining, you will be able to kind of identify roughly where your pluripotent population is and what you and where you should be isolating these cells and picking them from. Which brings me to our third step, right? In this case, once you've identified which colony you want to pick, we typically will go in and make these with either a needle or a sterile pipette. We'll make these scorings or these, cuts in vertical lines, right? Perpendicular lines across the colony as you see over here, just like so. And then we typically rotate our plate 90 degrees and do the exact same thing so that we create this checkered pattern, right? And the reason for doing this is you're actually trying to cut your colony into these little tiny squares of like or sheets of pluripotent stem cells. Now, this takes a little bit of practice, and in order to get your lines to be straight and not too wiggly, also to get the size of these squares to be, consistent and not too big and not too small. Because ideally, what you want to do after that is that once you've created those cuts, you can go in with a sterile pipette and very gently lift those sheets of cells and transfer them over into your next plate, right? In order for them to be passaged. Now, the reason why I mentioned that the size of these squares is important is if you make them way too small, the cells, as I mentioned, they really like to be together, right? They don't like to be singularized. They prefer an environment where they're in close contact with other pluripotent stem cells. So if those squares are too small, you're going to be stressing the cells out a lot. And when you passage them over, they may not attach or they may die, right? If they're too stressed. But on the flip side, if you make the squares too big, right, the square, once you plate it onto the next plate, might actually fold in on itself a little bit. And those cells in the center might start to differentiate, which will then, defeat the purpose of maintaining them as pluripotent stem cells, right? So again, you have to kind of optimize that size specifically. And eventually, right, you're going to, like I mentioned, scrape these squares with a sterile tip and transfer them over into a new plate where your media and your coating has already been done. Now, the traditional methods will use either needles, knives, or glass pasteur pipettes that have been pulled. But there's also, different tools that you can find out there that are a little bit easier to use, although they do come with some disadvantages. This is just one example with our StemPro Easy Passage tool. This is like a roller, if you think about it, with these thousands of needles in vertical like rows, right? So these needles are separated by a certain distance. And so what you could do is once you roll this onto your cells, you can create these perpendicular lines, like I mentioned, in one swoop. And then once you rotate your plate, if you do it again, you'll create these really nice, evenly spaced squares, right? Ready to go. But the downside of using a tool like this is it makes it very hard to pick exactly what part of your colony do you want to pick or not, right? So in an example like this with traditional methods, you'll see these cells right here at the very tip of the colony, for example, may not be cells that I want to carry over. So I may want to avoid, making another square here per se. But when using a tool like this, it makes it a little bit harder, right? To figure out which were really my good clones versus not, right? Or good cells versus not. So again, just things to keep in mind when picking your preferred method to passage your cells afterwards.
Omar, can I ask a question on that? This might actually be a naive question.
Please.
Are there automated methods now? Are there computer programs for this that like pick the morphology, pick the size of cells, that plug and play types of things? And maybe there are, maybe there aren't. But
That's a great question. I think so. And Kita, I'd love to hear your take on this too. But the field is definitely working towards that, right? On multiple fronts. So there are groups that are looking to use AI in order to take images of pluripotent stem cell colonies that have just formed and figure out metrics based on those images so that we can standardize what's considered to be morphologically good versus morphologically bad. Because right now this is very user dependent, and all stem cell researcher will tell you this, that different users have different propensities of picking clones or success rates of picking clones. So there is definitely a concerted effort. When it comes to the passaging itself, there are now tools that are specifically like colony pickers that are being developed right now. I haven't worked with one specifically, but Kita, I don't know if you have or you have any insight here.
Yeah, so actually we received the $4 million grant from California Institute of the Regenerative Medicine to establish the robotic automation system, collaboration with the Thermo Fisher automation team. Actually, our laboratory already have fully established equipment and for the iPSC reprogramming and the gene editing. So therefore, this one is benefit is that everything will be automated. So therefore, capacity of the feed and then also quantity will be like 100 level. And then for the colony picking, this is the really time consuming and the rate limiting stage. Just imagine if you pick 100 colony with this, and then someone just to look at the microscope for several hours, it will kill your eye. So, so therefore, what we developed is, as Omar said, we developed the AI-based image analysis. So therefore, automated microscope will take the picture, and then we trained the AI, and then AI will automatically mark which colony area is good colony versus bad colony area. And then just we trained the liquid handler, small the robotic arm, just mimicking the researcher's hand motion when we pick the colony and then move into the another tissue culture plate. So the benefit of the automation is that this one don't need a break, work 24 hours per day, even Saturday and Sunday. So therefore,
That's
they can pick the colony in 4 a.m. on Saturday morning. So that is the big benefit. And if you are interested in collaboration with like high throughput work, or also we are expanding the core-to-core partnership with some of your institution, and then you will see to access this equipment. Good question, Omar.
So no, this is it's great to hear, honestly, and I have to say I'm a little bit jealous because just to kind of give the group a little bit of insight, it it makes a big difference, right? And this is actually a great segue, Kita, into my next slide around, what do you do afterwards? Typically, you're not just picking one colony, right? We on average, right? We pick anywhere between 3 to 5 colonies per cell line or per patient that we're reprogramming or per sample that we're reprogramming, and we independently propagate them, right? So we propagate them as like clone one, clone two, clone three, and the reason for that again is these cells are finicky, right? They're not all of them are going to behave, not all of them are going to behave well. Some of them might differentiate, some of them might have some genetic defects because of the or like abnormalities because of the reprogramming process, right? And we don't necessarily want to use these. So we will pick multiple clones from each reprogrammed sample. And to Kitai's point, right? If you're reprogramming multiple lines or multiple cells or multiple patient samples, doing all this work becomes extremely time consuming. And let me tell you from experience, if you are able to lean on a method that is, able to do this automated anytime, right? Where you don't have to worry about coming in on weekends times, it's definitely something to consider. So I'll just say that. Um, but when you do pick your clones, right? One other thing to also consider is that you have to, we'll talk about morphology and things like that, but one thing to consider as part of the reprogramming is the Sendai clearance, right? So what do I mean by Sendai clearance? Can anybody in the chat kind of guess what that means? Let's see if we have no more lingering mRNA. There you go. Nice. Exactly, right? So with the Sendai vectors that we're supplying, right, to the cells, even though that it's an mRNA based virus, there's still technically some replication going per cell cycle, right? But these transcripts are not there indefinitely, right? This is a non-integrative method where these vectors are cleared over time, right? So most folks, what they'll do is they also want to make sure that their sample is free of this viral backbone, right? Or viral vector backbone, ensuring that we don't have those vectors in there anymore before I start using my cells, right? And typically this occurs with time. So as you continue to propagate from one passage to the next, you should see a decrease in the level of detectable Sendai vector backbone. Um, within your sample, right? But one other thing that I quickly wanted to mention is this process can be sped up quite considerably by doing subclonal expansion as opposed to bulk expansion. And so what this means is once you have a plate with a bunch of colonies, PSC colonies, taking one colony and putting that in one plate and maybe another colony here goes into another plane, not the same one here. So this would be like subclonal expansion, where one clone is picked out and progressively expanded over time. Whereas bulk expansion is you take all the cells in the well and you dissociate all of them, mix them in a pool, and then just split them by ratio into your next passage. And even though this, you still see you see the Sendai signal, trending downwards, right? In the 5 clones that we picked, we definitely see it going down with bulk expansion, but it's going to take a longer time. And ideally, with stem cells, because these cells grow indefinitely, right? If given the right conditions, we don't want to place too much of a burden on the cells when we don't have to. This is a really important concept that I hope we kind of by the end of this we drill into you guys. As sort of one of the core tenets of taking care of your stem cells is if you don't need to stress them out, try not to because the random mutational rate will still exist, but these cells can, like I said, under the right conditions, proliferate indefinitely. So they have a propensity of accumulating mutations over time, right? So even though, oops, okay. Right. Even though for the clones here, right, we can see that we've done some good work to pick the clones, show Sendai clearance, and stain for markers of undifferentiated cells like TR160, SSA4, OCT4, and SOX2, which by the way are by this point should be endogenously expressed by the cells. So these are even though that we had originally supplied these factors to the cells, the cells now should be producing their own because they have these genes that they were just turned off, right? And we can confirm this with multiple ways, right? With staining or with flow cytometry. But again, I wanted to bring this up specifically because assessing the the health of your cells or their genomic integrity is really, really important, right? This is going to be a topic that we're going to come back to over and over again during culture, after gene editing, where these cells are potentially stressed out is where we worry about them, right? And there's been documented research in the literature really explaining how pluripotent stem cells can really acquire these mutations in culture, and at a somewhat alarming rates for cells that again can propagate for again indefinitely or forever. And there's some work that was again published and also reconfirmed internally at Thermo Fisher, where we even see that these mutations in pluripotent stem cells tend to be in mutational hotspots, right? Maybe because of the way the DNA the DNA conformation during this state, but we very frequently find mutations linked to very specific spots in the genome. Gains on chromosomes 1, 12, 17, chromosomes 20 are the most common spots where you'll see mutations in those cells. And typically, the way most folks look at genetic mutation or in order to try to see if there's any genetic aberration is by doing these conventional karyotyping, right? Where I'm sure all of you have seen this in your traditional biology textbooks, these chromosomal spreads or these Giemsa stainings or G-band stainings where the idea is you have live cells, right? That you basically drop onto a slide and you allow the cell and you analyze the metaphase spread under a microscope, where your goal is to try to align the different chromosomes, knowing that you should have two of each, and compare the length of these two as they're stained with this like banding pattern from the stain, right? Now, the this protocol is sensitive to a certain degree in a sense of you're able to detect a mosaic population, meaning if even down to 5% of your cells had a genetic abnormality, you will be able to detect it because you're assaying 100 cells. So if just five of them are off, you'll be able to detect that. But in terms of disadvantages of these methods, these tend to be a little bit lower resolution, meaning that you can only detect very large gains or deletions because you're looking at, structural changes in the cells here, right? It does take some time to get these results because most of the time they're being done by a professional, in this case, a hemocytometer. I'm sorry, I can't speak today. By somebody who has the experience, but I'm a cytologist is what I meant to say. There are newer tools where there's spectral karyotyping, where instead of traditional stains, we're staining different chromosomes or parts of different chromosomes with certain dyes or certain colors that can then be referenced or measured digitally as well. But these still have that or similar limitations. And then the final one is that this type of assay does require live cells and dividing cells, right? So if you don't have access to a sample, your samples are too precious, you may not be able to do this for a while up until you do have the sample cells. Now, with that mentioned, one other thing that I will call out here is one thing that G banding does or traditional karyotyping does very well is the detections of inversions and translocations. So where you are able to, the same length of the or sequence of the gene is maintained, but potentially order or location is changed, right? So because we're viewing them visually in these cases, we might be able to see them as well. But again, you cannot detect like down to a single nucleotide change, right? So if there's like a point mutation, this assay will not pick it up. And as I mentioned, right, this is very important because we've done some work internally. We had a services offering a while back where we're able to take samples in and look at, mutations of these samples. We analyzed over 2,000 different samples from different folks. And again, we found that same pattern of these mutational hotspots in certain genes. But what was really interesting is that around 25% of the samples that we analyzed, and these were samples of, cells that people were working with, right, that had some sort of abnormality, right? And again, depending on what you're trying to do or the biological questions you're asking, you may actually want that, right? You may be wanting to model a disease of a particular patient. But if that's not what you're looking for, being aware of it is definitely important, right? And this goes also true for not just genomic stability, but also the pluripotency status, right? Pluripotency in a term is actually, I think, sometimes used or misconstrued because if you think about it, it's a functional assessment of these cells' ability to differentiate, right? So sometimes we say like pluripotency marker. Well, it's not necessarily a pluripotency marker, right? It's a undifferentiated marker related to pluripotency. I'm saying a lot here, but what I'm trying to say is that you have to look at how these cells not just look or their abnormalities, but also their gene expression patterns. Are they behaving like they should? And again, here we've done some work to verify that there are, certain samples that folks have been working with where there either could be borderline differently, have a different transcriptomic signature compared to what we traditionally know to be pluripotent stem cells. So as a precautionary tale, I always like to recommend folks to make sure that they're characterizing their cells every once in a while. These are cells that are going to be in culture from now on after reprogramming, right? And so maintaining that state is really, really important. And with that, I think this is my last slide. Just a couple of resources. We do have some e-learning courses that are free you can take that I've linked over here. The Cytotune reprogramming protocols, whether for research use or clinical applications, are also listed. There's also some reprogramming videos that could be interesting for folks that I've linked. And the final thing I also wanted to just call out here is I included a link to Shinya's Noble acceptance speech. I don't think I've actually mentioned this today, but that discovery was, Shinya's discovery, Jamie Sutton's discovery of being able to reprogram cells, right? Somatic reprogramming actually earned them the Nobel Prize, or at least Shinya's group in 2012, right? This is only a testament, I think, to the promise that this field holds, right? Because if you think about it again, this is then access to cells for indefinite renewal capacity in order to treat patients potentially with your own cells, right? If you're able to reprogram them from your own sample. And we'll talk a little bit more about that. But it's a super interesting read. I really, really highly recommend it. It talks a little bit about also how he came up with the approach and what they were thinking during this process as they were figuring out which genes are required, needed, sufficient, and or required to initiate this process. So with that, I'll open it up for questions and see if we have any. How are we doing for time? Yeah, okay, we're good. Any questions in the chat? Mycoplasma detection kit. That's a good question, Adley. We actually um again, I don't want to be too like forward here, but there are a lot of commercial kits available. If you reach out to myself or Tom, we can put you in contact with some folks. We have a mycoplasma detection kit that's QP. It identifies something crazy, like 160. Tom, let me know if I'm wrong, but like 160 different species of mycoplasma with a qPCR test that's simple to do.
Correct. Yeah.
Happy to send you information that way. Sweet. Anything else? Any other questions, Tom, that while I was speaking that you want to call out specifically?
No, I think we addressed most of them, and we had loads of questions today, which is great. Love the
Awesome.
participation, love the enthusiasm. So thanks.
Absolutely. Please keep them coming. As I mentioned, I know a bunch of folks reached out to me for some resources as well from last lecture. Let us know how we can help. We're definitely happy to. I'm hoping today wasn't too much of a bore and you have a slightly better understanding of what reprogramming is. Honestly, it's super cool if you think about what this means in the greater scheme of things, right? The fact that we can take a cell, pretty much reverse back time, and then get it to change in a way that allows us to use it for to generate any other cell type in your body. That's super cool to me. I don't know about you, but I quite like that. So with that, me confessing to being a nerd, I will pass it back to Kitai.
Okay.
And
Yeah. Great. Thank you very
yep,
much, Omar and Tom, for excellent lecture. And then also a number of trainee ask the slide and then the course material. And then if you look at the chat box, first one, I already included multiple linker. And then from those linker, you can download. So therefore, anyone just interested in this research material, and then go to the chat box all the way top, and then there is a multiple link. And then also another information is that genomic instability is very difficult because in average, pluripotent stem cell, about 5% always has unstable cell population exist. And if you provide any stress, and then those one will become a dominant population. So therefore, pre-count checking the genomic instability is really key factors. So that is the reason why is our center provide the whole genome, low density, whole genome based, what is that, genomic integrity analysis, CMB analysis. And with, what is that, $59.50. This one is almost a community service. We are not taking much profit. So therefore, if you are interested in, just contact the center. And then also we are collaborating with Thermo Fisher and then provide more details, genomic integrity analysis. So therefore, if you are interested in, and then contact Omar and then Nicole and then Tom, and as well as our center. And for the research material, we discussed with some official product, but this research material and the fundamental kinetics can be applied to other similar product from other manufacturer. Therefore, this is the very useful information. And then next week, we will cover the basis of the feeder-free human pluripotent stem cell culture, including key material mechanism and the chemical kinetics. And then while some example involve some official product, and then we will also discuss broadly applicable approaches. And then we will also have a guest speaker, and the Dr. Ming-Fan Ho from Mayo Clinic, who is famous with the creation of the mini brain in test tube. So this is the really, really good research tool. And then also Dr. Ho will present on stem cell clinical trial and her recent research on brain organoid, also called mini brain. And these concepts are broadly applicable across the tissue type. And thank you for see you next week. Thank you very much.
Thanks, everybody. Have a good week. See you guys next week.
Dr. Kim, the talks you mentioned, they will be part of this workshop?
Yes, because we have three special guest speaker.
Thank
And
you.
then this year we will discuss about bioprinting, AI, and then also the robotic automation. Okay, good. Bye.
Ciao, Kita. See you next week.